Okay, very good morning, students. Welcome back to lecture 6. Last lecture we discussed on next generation sequencing, basics of genome sequencing technologies. Lecture, I will talk more about whole genome sequencing. So the lecture has been divided into 4 outlines. For example, we will recap a little bit more on next generation sequencing NGS, and we have to go with a little bit more terminology here because we didn't cover as much as terminology in the last class. But this class will be a little bit descriptive on the terminology because that is very important for you to understand any genome sequencing or genome assembly. Then it will be taken into a whole genome sequencing approach through NGS, how the workflow happens, an example of typical Human Genome Project, how it was done through using NGS. That will be the outline for class today. So we studied in the last class different basics of DNA sequencing methodology. The one we covered the max last class was Sanger's dideoxy sequencing method. You should remember from basics that there is another method, Maxam-Gilbert method. You go and read back about that also. But mainly now the sequencing is evolved through Sanger sequencing only, the classical approach, individual method, individual sequencing. A single sequencing, you can sequence up to 500 to 1000 base pairs. And if you want to sequence multiple genes or multiple sequence, you have to go manually sequencing one at a time. That is a classical approach. Then we moved into next generation sequencing where automation happened, massive parallel sequencing also called as, and we are presently working into one of these 3 modules that's available in the market, but Illumina is one of the one that is doing a great job in the market. This single molecule DNA may not be necessary in the scope of the curriculum, but just for your sake of information, just look in that, we are going here. This is high throughput. It gives a kind of gigabases of sequencing, and it range of sequences 25 to 500 base pairs you can read. Those in a multiples of almost 1 gigabase compared to 1000 base pairs here. That is the high throughput technology we are in. Coming on to definition of next generation sequencing, NGS is a sequencing method where millions of sequencing reactions are carried out in parallel, increasing the sequencing throughput. This is the fundamental definition of NGS. If you are getting into a question marks of 2 mark, 4 mark questions in the exam, that you should be able to ready to write this definition. And if it takes advantages, what are the advantages of NGS method? You go into this abbreviation. The NGS method uses array-based sequencing, which combines techniques developed in Sanger sequencing to process millions of reactions in parallel, resulting in very high speed, high efficiency, high throughput, and with a reduced cost. That is advantages, a definition here. So we spoke briefly about different evolutionary trends of sequence methodologies. For example, starting with 1970s, you start a proto-sequencing, you are able to sequence single small stretches of genes or DNA. Then you went into manual sequencing of generation 1, generation 2 automated Sanger, then generation 3 that don't talk about clones, that talk about clusters. That's coming in terms of Illumina and Roche, and then single molecule, that is output in terms of nanopore, bio, and helicose. This is may not be necessary, but try to understand of generation 3, what are the advantages we came across. This is a glance of looking into the data and publications or in terms of research work you have been observing. A typical proto-sequencing data looks like this, very small reads. You're looking about into a kind of 4 nucleotides only here, 1, 2, 3, 4 only. It was amazing to see this data at that time. 4 nucleotides, they were very happy. It was published in a prestigious journal. How they analyzed it's old-fashioned technologies they use, agarose gel and probing, blotting technologies, and radiography technologies they used. Then you move into Sanger sequencing, where excuse me, you use the agarose gel electrophoresis. You try to identify based on the size what are the nucleotides based on the ending of the reaction with dideoxynucleotide. That moved into technological revolution. You went into automated sequencers, which are now from single automated sequences to capillary electrophoresis here, reading the reads. You don't need to do multiple gels to understand. You can run through a process of machine. that can run through samples and read the output at the chromatogram. Then it's not chromatogram. It's another definition. We will see. It's not chromatogram. Chromatogram for chromatography. So then it consists of clusters in the Illumina generation 3, then single molecule. But clusters is important. You see that you can see millions of pools of sequences there. So again, we looked at comparisons of Illumina, 3 different systems here. NextSeq 500, HiSeq 4000, NovaSeq 5000, and one more 6000 is there. These are the machines Illumina has developed over time, and each machine will have a capacity of advanced and technological capacity that goes up here. Okay, anyone doing a chat box? I think, hope everything is fine. Okay, so you can look into the terminology here. The output range from GBs, you are looking from 120 GBs to 6000 GB here. Runtime, you can see from days to hours. You go from days to hours here. And the reads per run, max read length. This is really important. What are these terms? What are the reads? Read length, max read. What are these in terms of NGS? So this class covers all the descriptive terminology for that. Okay, we got into the glitch now. We'll stop share and then go back share again. Okay. Then come back. That taking from the Human Genome Project, we started in 1990. Basically, it started with basic technologies in sequencing and growth gel electrophoresis. Then in 2001, it was completed. At that time, the cost per megabase of DNA sequence, megabase, you're looking at the megabase, 1000 bases of DNA sequence. It was 10,000 US dollars at that time in the beginning. But you see in the trend in 2019, it come into 1 cent, in the range of 1 cent. So basically, the price reduced by 1 million fold over 2 decades, 20 years. That was the amazing thing we have to see, that economy in terms of reduction in the price allowed us to sequence more and more genomes in the long term. So coming on to what are the steps in the NGS method, what a typical next generation sequencing, if you want to go for a whole genome sequencing, for example, a bacteria, yeast, human, or for example, coronavirus, COVID-19 causing organism, you have to go with in terms of the typical steps that constitute the main foundation of NGS sequence. The main steps are input DNA, library preparation, sequencing, and then data analysis. These are the 4 steps, the fundamental steps that any NGS sequencing involves. The input DNA, typical any organism will have genomic DNA. Sometimes viruses will have RNA, but basically typically genomic, and then genomics consists of DNA. Then you can put the DNA into that. But as you know that the genome varies from species to species, organism to organism, species to species, and it is confined to in the nucleus in a 3-dimensional structure, and it's already in the nucleosome, it's packed. So you need to have a methods to isolate the DNAs, and basically you cannot put into the machine one input DNA of 3.2 billion base pairs and switch on the machine, go back in 2 days, get the complete sequences here, and then you go into analyzing the data. It will not happen because the machines are not robust enough. They may be robust enough, but you need to understand one more fundamental process, that the chemical reaction that happens in synthesis and the enzymes, they are error prone. They do errors because you know already that DNA repair mechanism, even the DNA polymerase has fidelity problem, but it's highly efficient. But 1 in million base pairs, it will give a wrong base pairs code. It attaches the wrong nucleotide. For DNA polymerase, that is the fidelity problem, but we are all doing these reactions in vitro. So imagine that a chemical reaction that can cause us a lot of errors in a given time. So it is highly impossible to sequence whole at a time, but if you go into long stretches also, there is a fidelity and error problem. So you go into a specific steps of library preparation, you cut the genome into small pieces, store them in a certain system, then you put them into sequencing that, and data will be analyzed through cloud computing. A lot of bioinformatic tools will come up. So coming on to in few slides, we will talk in terms of a technical jargon for NGS or terminology, then go into typical one example, and then go into human genome sequencing, how it is done. So what are reads in terms of NGS? The output of an NGS sequencing reaction is called as a read. One reaction or a typical multiple reactions or parallel reactions. You take one reaction at a time, one NGS sequencing reaction will give one read. Like that, it gives multiple millions or billions of reads. A read is a single uninterrupted series of nucleotides representing the sequence of the template. Because you have given the genome, you have given a DNA, so there is a template genome already there. You have a primers in this direction, read 1, read 2. You start the sequencing reaction, it goes and completes up to this. This read goes and completes up to this. So this will be one read, this will be one read. So read 1, read 2 for the given template. Like that, there will be multiple reads. For each typical sequence of the template or typical nucleotide, there may be multiple reads. And the typical read length is called as the length of each sequencing read. For example, if this reaction is complete, you may have a 10 base pairs, 100 base pairs, 1000 base pairs, 10,000 base pairs. Then accordingly, the read length is ranges to the whatever completion uninterrupted nucleotides here. Typical sequencers produce read lengths in the range of 100 to 500 base pairs only. Please remember that 100 to 500 base pairs only. Why? Because the enzymes may cause wrong coding that causes an error in the sequence. So to avoid that, you better don't go with the read length typical of 500 base pairs. But again, there are efficient mechanisms that may allow, but mainly recommended is 100 to 500 base pairs. For example, Pacific Biosciences in generation 4, it may go into read length of 1500 base pairs. There may be errors, but you don't want to spend too much time on going to short reads. You can do in one step 1500 base pairs. If errors are there, it's okay. I can deal with the reference genome. I can annotate it again, but I just want to get the data quickly. You can go ahead for this, get and annotate the data. So read length is always average. For example, given a typical nucleotide, the read length can be varied. I will show you in the next few slides. It's all usually taken as the average read length for each given an individual reaction. We spoke in the last class, single end reading, paired end reading. Single end reading going from one read to direction to 5' to 3' only. Paired end reading is go to one direction, again go from 3' to 5'. This single end is typically used for small genomes or prokaryotes, or probably the RNA sequences that usually get small, RNA genome sequencing. RNA sequencing, and paired end is used for whole genome sequencing. And also the length of the reads is called short reads and long reads. Short reads are usually, you see, this is a reference sequence. The reads that the reaction can give is a very short, up to 150 base pairs. It's a multiple of 150. One of the Illumina system automatically uses that. You can see small reads, long reads. PacBio and Oxford Nanopore, this generation 4, they will have a typical length of long read close to 1500 base pairs. And this is for simpler genomes and prokaryotes. This can be used for eukaryotic and complex genome. And when you have a close look at here, you're talking about reads in this lane, 1, 2, 3, 4 lanes, 1, 2, 3, 4 lanes. You take a typical example of one nucleotide sitting here. This is a stretch of ATGCs, right? You're talking about. Then you're talking the same sequence here with multiple reads. For example, you can see that there, let's say there is a C here in this stretch. You go into reading in that, it can be caught in the 4 reads, all 4 reads can catch it. But the A sitting here, it can be caught into only one read. And also you should remember that read 1 here in lane 1 is overlapping with the read 2 in the lane 2. And everything represents again to the same basic template DNA. It's not a read, lane 1 is not different from lane 2, lane 4 is not different from lane 1. All of them within this end to start to end represent the same template. There is a varied, there is multiple reads for each nucleotide or each sequence. So you see that there is an overlap. You see here a bigger graph, you can see an overlap bigger here. So read 1, read 2, read 3, read 4, lane 1, lane 2, lane 3, lane 4. You see there is an overlap. There's an overlap 5', 3', 5', 3', 5', 3'. So when you try to stitch this length here based on the reads, you combine all 4 reads together of this point to this point, you get a single stretch of assembly. But you can see there is contiguous overlaps within the reads. That's called multiple reads do overlap contiguously along their read lengths. This you need to remember. And the terminology now comes, these are called contigs. Contigs of in a terminology in NGS is a set of overlapping DNA sequences, a contiguous piece of DNA sequence that has been assembled from more than one read. For example, a contig here, reads are here. It is compiled from these reads because 5' 3' sequences of the another overlap. You take the overlap, you compare, you come to a consensus, you draw the contig 1, contig. And these are scaffolds. That's one more, you don't need to work on that. You make a scaffolds of that. The genome, reads, and contigs. Look into that. So then comes in terms of coverage. The number of times a particular nucleotide is sequenced. For example, we are talking here, let's say length of the DNA, 80 GCs randomly are there. One example, I think I have another slide that explains. So you want to see this particular nucleotide of the 480 GCs, how many times it has been sequenced given a same template strand. And coverage, again, why you need multiple reads? Because error-prone sequencing reactions, random errors could occur. Then along the 5 reads, you see, if there is no match that it should read for A only, all the 5 reads should show A. If one shows T, then the efficiency is less for the read. So that's why you go into 30x coverage typically. You sequence each nucleotide 30 times and then you do multiple reactions, right? So 30x is a typical to say on a consensus, but you can go from 10x to 100x is also a typical coverage here. The number of times a particular nucleotide is sequenced is called coverage. And deep sequencing, why 30x only? I want to go into more than 30x because given a sample of my genome has a high polymorphism, high variability. For example, my genome is different from your genome. We differ in terms of single nucleotide called SNPs and indels. We will talk in coming classes. So if I want to compare my genome with you along with the reference genome that already sequenced, we need to go into a sequencing machine, give my DNA, give your DNA, start aligning it to the, compare it to the reference genome and see how you are different from reference genome, how I am different from reference genome. So it's actually not by mistake, it's actually variability from individual to individual. So you need to go for deep sequencing. You may go for more than 100x or 200x, so that how variably I am different from you. Consistently, A in me in a sequence differs in a position, you may have T there. So you have mutation in yours, variability in yours compared to me. So to do that, you need to go for deep sequencing. Deep sequencing increases range, complexity, and accuracy of the result. It is used to study rare polymorphism between humans that expresses a mutation, for example. Or in a cancer polymorphism, you need to identify cancers. So again here, you are looking for a typical chart here, depth of coverage, for example, reference genome here, depth of coverage and breadth of coverage. You are looking all along the length of the genome. You are looking how many times you read it. That coverage is 1x, 2x, 3x, 4x, and 5x. A typical example, let's say A sitting here, I might cover it only one time in these 5 reads. When I did 5 reactions, I could able to see A coming up in only one reaction. So I can say it is 1x. But if I say typical stretch here that can cover 5 times, then I say 5x coverage, that may be T here. So I covered the T in 5 times. In 5 reads, it's always T. I put a tick mark in that, this position T, I'm fine with it. At the same time, you may find in a reference genome, you may find a read that may not cover it at all. There may be C here. In your reads, you may not get it. That may be called as 0x read, where you are unable to read at all. It may be one nucleotide, it may be gaps. These are the indels will give a lot of gaps on 20 to 10 to 20 bases. So you may get a 0x also, remember that. So coverage 1x to 5x. Here the uniformity of coverage, you are going again reference genome, a reference of GRCH37 reference of human genome or some other genome here, reference genomic strand, NGS reads. You go in to see that the more reads, 10 reads here, you have 10 lanes. And you're looking at the nucleotide here, you can see that you are sure it covers in all that. So you have a coverage depth of 10. coverage depth of 3, coverage depth of 2, and it can be a depth of 0 also. So that is the importance of depth of coverage, where you can see the multiple reads. So basically, it brings that. For example, this is an example of computing sequence coverage. So whatever you're doing here, you put in the NGS system, you can look into stuff that you're looking at a clusters here. Clusters means you're looking at generation 3 sequencing. You're looking at a pair end, so you're sequencing on both sides. The read length is 100 nucleotides, the machine took it. So the total human genome is 3.2 GB. So you're looking at a formula, number of clusters into pair end into read length by total genome. You come into a point of 12.5x coverage. For example, of the 3.2 billion nucleotides gene or nucleotide sequence of human genome, this machine has covered it that every nucleotide was read through 12.5 times. So that is the coverage of. So it covered whole 3.2 multiple times of each nucleotide, that goes into 12.5x coverage. So the coverage is directly proportional to the read length or number of reads. Number of reads or read length, not read length obviously. Number of reads is important. The more reads you get, for example, 10, 12, 50, 100 reads you get, higher the coverage will be. So that's a general rule. And Human Genome Project in 2001 was covered in terms of coverage was almost 9x. Each nucleotide was sequenced up to 9 times. Okay. So again, we need to speak into adapter molecules. This helps in another sequencing methodologies. An adapter is a unique sequences used to cap the ends of a fragmented DNA. You cut the DNA into pieces, then they may go for complementarity again, right? Then you don't know which cut you have done. So you put an adapter to that, to the ends, then based on the adapter sequence, which you already know, you can easily pick the DNA. So adapter helps allow hybridization to solid surface, also a glass surface, provides priming location for both amplification and sequencing of primers, and it provides barcoding. It also, you can code by barcode. It makes a simplicity when you're looking multiple sequence reactions in parallel. That we discussed in the last class, the typical barcode of adapter 1, adapter 2, you put it into genome clones, ligation happens there. Again, you go into denaturation, single strands of the DNA will come up. Then you go into assembly, where a reference genome here. The contigs reads are 1 here. This may be a contig, reads there 2, contig. You can easily identify the assembly there. So again, coming on to adapters, that's one thing. But again, you need to remember a term of library. So your genomic sequences can be put into libraries, because you are tearing it up. Then you are tearing up in small pieces into short and long reads to make advantage for the system. But you will have billions and billions of pieces. Then how do you make? You need to make a library of it. The library can be done with adapters. A collection of DNA fragments with adapters ligate to each end. Library preparation is mandatory, is required, is necessary to go into any sequencing run. Otherwise, you get you cut the DNA, you put into the machine, it gives something at the end, but you don't know what you're looking for. Though you assemble the genome, you have no idea what you're looking at. And there are typical libraries called genomic and cDNA libraries. For example, here, DNA library construction, RNA library construction. You know that the genomic DNA is complete a kind of 3.2 billions in one unit, right? In a cell, the genome is compact. But central dogma, genome is into RNA, RNA into DNA proteins. The RNA consists of small pieces, right? mRNA molecules, tRNA, some other molecule. It's not a complete stretch. It's already a small fragments of mRNA are available. So you know that mRNA sequences of the genome, the total genome. For example, DNA library, you go into fragmentation of the DNA, then you may go into PCR to get amplicons, but fragmented DNA can go into adapters. The sequencing library will be generated, adapter ligation and PCR. Already you have RNAs, you go with RT-PCR. We already know that reverse transcription, RNA to cDNA, RNA to DNA is called complementary DNA, that is reverse transcription. Go with RT-PCR, you make cDNA, adapter ligation. Then you go into sequence library for RNAs. Why not, why cDNA, why not RNA libraries on its own? The green can go into that. But the problem is, Compared to DNA, RNA is highly degradable. It is susceptible to RNA's degradation, contamination problems. If you make a library, you're not sure whether it's going to sustain or not, it's stable or not. So better you go into a complementary DNA so that lifetime of the library will be enhanced. Quickly, few more terms. That alignment, mapping of a sequence read to a known reference genome, that's called alignment. Reference genome already you know for humans. That is usually called as a fully sequenced and mapped genome used for mapping of sequence reads. So it is fully sequenced and mapped. We will see the terms in Human Genome Project. And alignment, you align with the reference genome. And one more term called de novo assembly. For example, humans, we have a reference sequence that is sequenced, reference genome. Let's say if we don't have any reference genome, we are starting with a novel genome, isolated organism in, you went into for C, and then you found out some protist or some sea-dwelling animal that no one knows about it. So you sequenced it. Excuse me. Anora, give me a minute, Amma. So that's called de novo assembly. De novo assembly, it's a little bit complex. The methodology requires complexity. It needs like kind of gene annotation tools, gene function tools, those kind of things are. It takes a bit complexity, but if you have a reference genome, it makes simpler. Then specificity and homopolymers you can look. Okay, that brings back to a point where we discuss different aspects of NGS, different terminology that is fundamental to better understand how the NGS system works. So go through that. It takes some time, and already the description is here. It makes easier for you to look into and refer it back. And we will take now this example of NGS. We go into understand our human genome, how human genome is sequenced. Let's say Human Genome Project, why we did it, what we did it, at the crux, what is the crux of it is that it's nothing else. You know that there is a blueprint of our body, the blueprint of any human or any organism resided in a cell. And you know the length, typical length varied from species to species. And we know our length is 3.2 billion base pairs. What is the Human Genome Project is that I want to know the code, nothing else. I want to know the code, and we know that the code is not binary as 1 zeros for computers. The code is having 4 units, ATGCs. In computers, it is only binary code, but it is quaternary code here. So in one sentence, we are trying to decipher every nucleotide of the 4 ATGCs within the 3.2 billion base pairs in order. Starting from the first, I want to write a book on a human code, human genetic code. I just want to write all the 3.2 billion base pairs in a book. I want to start with the first nucleotide in our genome to the very last nucleotide in the genome. I just want to keep writing 3.2 billion bases in a books. How I can do that? That's the main essence of Human Genome Project. Why it's taken, what advantages, we will see it later. In one sentence, this is the intention of the Human Genome Project. So there are some facts here. You need to understand the basics and ethical and in terms of how humanity came into multiple diverse fields with one goal, and it's a very appreciative or unexplainable task. No one would imagine that you are going mad. Initially people that hey, I want to sequence my code or human genome code. People would laugh at you. What are you talking? Is it possible? It's written by God. How can you code it? But it's an amazing feat. Humanity come together all over the world, different scientists, and they achieved it and they succeeded it. It's magic, to say in one word. So whose DNA did we sequence as reference sequence? So for the Human Genome Project, usually the samples collected for blood from female samples. Anonymously, 100 people donated samples, and sperm is used for male samples. A large number of donors, but of them 100 are there. Of the 100, few are picked, and this anonymous. Donors are which sequence has been taken, no one knows it. Only few samples were processed as DNA sources, and the source names remain confidential. Neither the donors nor the scientists know whose DNA is being sequenced. Just like they given a sample with the numbers, you go ahead with sequencing, that's it. And it's done by a company called Celera Genomics. It's a combined effort of different branches, different scientists without any political borders. Whole world came into one stance, and the draft genome sequenced 99.99% in 2001 and completed in 2003. How much money it cost? A few other terms, and you can go through the text here, the facts for human genome. So let's take an example. I want to use NGS, go with doing human genome sequencing project. There will be different steps. So the steps starting from the 12 steps I have shown here, we will see quickly. So start with the genome. You go with physical and genetic maps, cloning, subclones, storage in E. coli or bags. We will see what are bags. And then preparing DNA for sequencing reaction, products of DNA sequencing reactions, separating DNA fragments and reading. And parallel you read, again you separate it by gels, Kepler electrophorese. You assemble it again back, you assemble it, and then you refer it back, then the working draft sequence comes, then the complete draft. These are the typical steps we follow. And where do we get the human genome samples? We already know reference genomes are there, but if you want to sequence my genome or your genome, how do you go ahead with that? But remember that typical cell has 1 or 2 copies of the genome. If you are diploid, we are diploid, then haploid, 1 genome copy only, diploid 2 genome copies. So you are taking, I'm saying that, okay, isolate whole genome from 1 cell and start doing that. It may happen, it may good for some other reaction methods. For example, single cell is fine, but in general, you need lots and lots of starting material, that is the genome DNA. So you isolate it from 1,000 or 100 times of 1,000 cells. Basically, human body has 100 trillion cells. So you take few set of examples, isolate the genome from those cells, isolate the DNA. Then it's also important that you should remember that you are not going as a de novo assembly, you are going with the already known human genome. And already one of the advantage for us is that we know physical and genetic maps of genomes. For example, the gene that codes for eye development, where is it? You know it, it's on chromosome X. What are the gene that goes for limb development? You know it, it's there in the chromosome 10, or for example. So where is it located in the chromosome 10? You know it already. So we have a landmarks, the physical and genetic maps of the human genome. These landmarks will help us during assembly. So you know that where you are looking, this stretch matches to the landmark here, physical and genetic maps. Then you know that you are trying to catch from this reads, millions, billions of reads of sequence, you're specifically locating to one spot. The definition of genetic mapping, physical mapping, you can look up here. This is a typical example how it happens: chromosome, genetic map, physical map, DNA sequence. You are looking at UCSF genome browser. The typical chromosome 11, you are looking at length from starting one code, one DNA to another DNA. That comes 9,000 base pairs. You can go to the stretch. You can see the short and long reads here. What are the genes here? It's already an assembled sequence. You can go through the genome browser. Again, there are different steps as we talk only breaking the genomes into long and short reads, then cloning, then bacterial clones, and we will see in yeast artificial clones next class. But you clone into bacteria and you clone into bags, you clone into reference sequence, now you can study. This is already genomic DNA for reference sequence assembly, but you want to study a new human being or myself or yourself, you again go through the same steps, you just compare it to the reference sequence and identify. There are different methods that can come across here. Libraries, bags we will see in the next. Clone by clone approach, clone contig approach, shotgun approach. In terms of, we said we will clone the broken genomic sequences, right? You need to break the genome into small pieces. There are different plasmids we can use. One of them is called bacterial artificial chromosomes or bags. These are, they can store up to 100K or 200K DNA sequences. You can do 3 same method fundamentals. And also it's important that we are looking into typical, you are storing the DNA in small pieces in the clones in bacteria, or bags are usually bacteriophages that attack bacteria. You're looking bacteria and bacteriophages. How do you grow them? Typical plate here. You can culture them in the broths. So when you are doing NGS, you are looking into a robotics and automation. You have plates, they have automated to pick a single colony at a time based on the location. They are able to take the colony and broth it so that there is automation, manual labor can be avoided. Then you go into specific typical attaching typical sequencing reaction. Next few slides talk about shotgun and hierarchical shotgun. You go through the steps here. Basically, you're hitting the DNA shotgun. So you break the DNA into random pieces, then you assemble it here. That's a terminology used for shotgun sequencing. You go with the same steps we have discussed. Go through the text here. You will better understand the process. Again, this is a shotgun approach to talk about mapped clones and whole genome shotgun approach. You are looking into a physical and genomic maps here. There's one more slide that explains the text here. You go through that. Some more terminology. You can understand the definition. What is the BACS? You can see through that. And it comes to an assembly. There is a terminology that what assembly N50 I am looking, and what is the version of it, what is the specific name I am looking. And you can see that there is a 2004 assembly, there is a specific ID given, and the assembly will follow separate steps: input, raw data, FASTQ, different steps. You go with the assembly system. Again, go into that genome, raw reads from the sequencing machine, quantic assembler, quantic FASTA format. You go into analyzing which are the genes, protein coding or RNA genes, and process. So that brings back, I think the timeline, you can go and understand it a little better way, but it is a typical assembly reaction that fast.